This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 434, recorded on March 21st, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase and free shipping by going to blueapron.com/TWiV. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in a sunny New York City, Dickson, Despommier.
Good afternoon, Vincent.
We are recording at an unusual time of the week, a Tuesday.
We are.
Due to scheduling constraints, but it's a very pretty Tuesday.
This half-weekend virologist.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
How does it feel for you to be a Tuesday in TWiV?
It's disorienting.
I'm sorry. I'm so sorry. I know.
No, that's okay. It keeps me on my toes.
You have nice weather?
It's okay. There's a sort of a high broken layer around 7,000 feet, but other than that, it's 12C. Wind is not too bad. So.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. We have the same temperature as Alan. That means it's 54 degrees Fahrenheit.
Someone wrote on the blog at microbe.tv/TWiV, the latest episode. He wrote, "Can we just skip the weather and get to the virology, which is what we come for?" And so I'm answering, no.
Right.
If you notice, most people who write letters end up telling us about the weather.
That's right.
So what I would suggest to the writer of that comment is, why don't you skip the weather, and we won't.
We could begin by saying 2 weathermen walk into an isobar.
Oh, that's good. And what happens next?
They got under the weather. I don't know what.
Yeah. They were under a lot of pressure.
They were. Thank you.
So we're recording on Tuesday because I'm going to Denver on Thursday for 2 days.
Mm-hmm. Have a steak for me while you're out there.
No, I'm not going to have a steak.
They have good steaks out there.
I understand. And today I just returned from Rockefeller University, your old stomping ground, Dickson.
Yep.
Where I taught virology class.
Well, you told me that it's changed quite a bit since I've been there, though, haven't you?
Yeah, it's changed quite a bit. Anyway, this class, it's a 2-hour lecture. I gave it every 2 years, and I've been doing it for many years since it was taught by Haizaburo Hanafusa. No, I don't give the same lecture. It changes every year. It's on coronaviruses. And then we have a discussion. I assign 2 papers, and two students in the class present them, and we sit around a table and have lunch. And I have no voice as a consequence.
You have no voice.
No voice, as you can hear.
Sounds pretty good to me.
I am laconic today.
I
love that voice.
Isn't that in New Hampshire?
That's a parkway upstate, the Laconic Parkway, isn't it?
The
Laconic.
Do you know the Laconic, Alan?
That's the Taconic. There is a Laconia, New Hampshire.
Yeah, there is.
There is. My sister used to live there.
Oh.
The American Society for Microbiology is putting on Microbe 2017, June 1st through the 5th in New Orleans, Louisiana.
New Orleans.
The birthplace of Dickson Despommier.
This is true. And others, too.
Well, you're the only one that matters, isn't it?
No, I always look at that as the birthplace of Louis Armstrong and jazz as we know it.
This meeting will provide a unique platform to explore the full scope of the microbial sciences. ASM Microbe 2017 will feature an exciting program with opportunities for you to gain great new science and connections, whether you're a researcher.
Is that the full microscope?
Yeah, it's microscope, exactly. Whether you are a researcher or educator, a clinical or applied microbiologist, an infectious disease or environmental microbiology expert, or even if you're retired, explore the complete spectrum of microbiology at ASM Microbe 2017. I added that part in, the retired part.
It was fine.
Register before April 20th and save. For more information, asm.org/microbe.
You could submit your too late breaker if you'd like.
That's a good one, Dickson. Are you in a good mood today or something?
Actually.
You are?
Yeah.
Why?
No particular reason. Just
Some days
all
she
the
doesn't need 'em.
systems
It's
seem to
good.
line up okay today.
Have one follow-up, and I'm not surprised because it's only Tuesday, and probably no one's listened yet. Well, some people have. Dear Vincent et al., just like a paper, huh? As a longtime fan of TWiV, it was a real delight to hear our work on super spreader phages profiled in a recent episode, TWiV 428, Lice Globally, Protect Locally. Thank you for choosing to highlight our paper and, as usual, for superbly explaining the underlying science and its implications. With best regards, Eric Keene.
Oh.
Now, where was he in the paper? Does anyone know?
I'm sure it was someplace nifty.
Oof.
I was actually just in Keene this morning.
Keene.
I was in Keene.
How far? That's New Hampshire, right?
No, New York.
Yes.
Keene is the first author.
Could
Probably
be New
they were
York, too.
co-first authors, right, Kathy?
Yes.
I'm looking it up. It's a ways back though. It's not
a little while
exactly
ago.
follow up. Yeah.
No, I
Right.
put it in because there was no follow-up.
Oh.
And he's a first author. Address correspondence to Eric Keene at the University of
Yeah.
Miami. Wow.
Yeah.
He's the first author, and he's now in Miami. Or maybe he always was. Let me see who the affiliations were here. Oh, how do you do that? Author.
We forget things immediately afterwards.
University of Miami, NIH,
Yeah, he was there. He is there.
NCI,
Cool.
Super Spreaders.
Fort Detrick.
It's a cool paper. You're welcome, Eric. No
And
problem.
thank you, Eric. That was
Yeah, I know it's
work.
not follow-up. It's technically defined as from the previous episode.
right.
But I thought it merited it, and it's very
Sure.
short. Now, we have a paper today.
We do.
We always do, right?
No.
We haven't had
sometimes
any-
we've had a mail. Sometimes we've just read mail.
We
And
haven't-
sometimes when we have a guest on, we don't do.
Oh, by the way, speaking of paper, I meant to tell you. I forgot. There was something from last week. Darn. What did we do last week? What did we talk about?
TWiV 433.
Thank you, Kathy.
Well, and when I pull it up in my app, it's not going to give me the little summary because TWiV is
It's broken.
glitchy on the...
Poops.
Right. That was poops, viruses, and worms. So that was
Right.
the fecal microbiome and the worm.
Oh,
That's
okay.
right.
Yes, I
Yeah,
know.
that's right.
And as I say, I wake up at all hours and I think of things I should have said on TWiV and I didn't. So here it is. We're talking. They didn't find any eukaryotic viruses in these sampling of poop, right?
Right. Yes.
And you, Dickson, said, "What would you expect?" And I said, "Oh, well, herpes viruses, adenoviruses." And I forgot to say enteroviruses, rotaviruses, noroviruses, but Coxsackies, enteros, all of those are actually plentiful.
But don't you have to be sick to have them coming out?
No, not at all.
Not a lot of them.
Here's an
But-
interesting.
So you become a
But
carrier
wasn't
for
the-
these things?
Wasn't the paper only looking at DNA viruses? So that's
right.
maybe
DNA
why you
virus.
didn't think of it.
That's absolutely right.
Right.
I was thinking, but there should find, among the DNAs, adenos and herpes and what else? What other DNA viruses? Probably polyomas too, right?
Yeah, and let's see, is Circo a DNA?
Yeah. If it's circos, they should find. Okay. But RNA, there should be a lot of RNAs. And the reason I remembered is because when kids get the polio vaccine, the Sabin vaccine, after a few days they excrete revertants, right?
Right.
Right.
They're all recombinants with enteroviruses in the kid's gut. Yeah. Every one of those revertants is a recombinant.
Wow.
And so we must be populated with enteroviruses. Okay, that's what I wanted to say. The other thing was, today at Rockefeller University, by the way,
Yeah, rock you.
we did 2 papers, which were my choosing. Of course, I picked 2 papers we did on TWiV.
Again, killing 2 birds with one stone.
I mean,
Yes.
Right.
I figure I vet a lot of papers for TWiV, and why not just pick one of those? Why do I have to vet all over again?
Sure.
You don't want to vet more than once.
Not you bet.
And we did that last paper that we did where they did the big haploid cell screen, and they found a protein that helps picornas get out of the capsid, and then another that tries to direct them towards autophagy pathways. Remember that paper?
Right.
I did that. And then I did one of Bert Semler's papers on the unlinkase.
Oh, that's nice. Yeah.
Yeah, we did that. And too bad Bert wasn't there because they had all kinds of questions. Like, why didn't you do this and why didn't you do that? Okay. Now to today's paper, PLOS pathogens. Senator Eisen, oh no, he's not a senator yet, was the founder of PLOS, among others. Harold Varmus, right? Yep. A novel negative-stranded RNA virus mediates sex ratio in its parasitoid host. I got that title now. I understand it.
There you go.
Especially the negative-stranded part. The first two authors contributed equally to this work, Fei Wang and Xi Fang. Do you pronounce?
And it's symbolized by a yin-yang symbol.
Oh, that's lovely. That is
a little
great.
icon of yin-yang, yeah.
Do you think that was the author's choosing?
I don't think so.
The journal editors? Maybe it was maybe it was Lily.
Yeah, maybe.
Hey, Lily. Do you pronounce QI she?
I do. I don't know
chi?
if that's correct. Chi.
Yeah.
All right. And then the other authors are Wang, Yang, Hong, Bao, and Jens Kuhn. Wow. Where did you come from? Oh, John Warren, Song, and the senior author is Gong Yin Yi. And this is from Zhejiang University. And it's from NIH, of course. That's where Jens is, the Integrated Research Facility, University of Rochester, and the University of Missouri in Columbia, Missouri. Do you pronounce the I, Missouri or Missouri? Okay.
Missouri.
I say Missouri, but I'm not from there.
Right. Show me.
Parasitoid wasps. Let's start with that.
Yes.
Shall we?
What is a parasitoid wasp?
Who wants to
Dixon,
answer that one?
You don't know, Dickson?
No, I know. And I know that Alan knows because he's got a rich history.
Go ahead, Alan.
Parasitoid wasp is one of the many, many species of wasps that lays its eggs in usually another arthropod host.
Mm-hmm.
If you saw any of the Alien movies, you've seen a Hollywood version of this.
Right.
They tend to they often parasitize Lepidoptera.
Yes.
And I guess they're called parasitoids instead of parasites because they're not microbes. So it's an arthropod laying its eggs in another arthropod. Frequently, they will paralyze a caterpillar, lay their eggs inside it, and then the eggs hatch. The larvae eat their way out through the live caterpillar and pupate and then fly off.
Yeah. I like the one that catches tarantulas and then lays its eggs inside the tarantula.
Right.
Imagine
as if you
confronting
needed to
something
make
4 times your size and you win anyway.
All right, so viruses of 7 different families have been identified in parasitoid wasps.
Right.
There are
and we've talked about some of these.
Yeah, especially the Polydnaviridae, right?
Right.
The double-stranded DNA viruses. There are also plus-stranded RNA viruses, coronaviridae, eflaviridae, and also double-stranded RNA viruses, reoviridae. We've talked about the polydnaviruses because those are the ones where the capsid is encoded in the wasp genome, right? And the wasps produce virus particles with some genes in them that the wasp then injects into the caterpillar along with the eggs, right?
Yeah.
And then the viral genes are expressed, and they prevent the caterpillar from basically eliminating the eggs, right?
Encapsulating them. That's right. It inhibits cell fusion.
So the cool thing there is that the wasp, in its genome, has the viral genes.
That's remarkable.
I mean, you cannot invent nature.
No.
You know, all the stuff that happens, you can't even make a movie because the movies don't even come close.
No.
And that's
Alien was way off base in some biological senses.
They should remake it with CGI instead of those crappy special effects they had.
I don't know.
I really liked.
I liked those special effects. Actually,
yes,
they were pretty
I
nice.
Really liked the original movie.
The first one was good because they hardly showed the monster and it scared the hell out of you.
That's the whole key to monster movies.
Yeah, yes, you're right.
It's amazing.
Oh yeah, Kathy, did you see Alien?
I did not.
Oh.
Yeah. It's a gap in my knowledge.
We had a wonderful session at the American Society for Parasitology meeting
TWiV,
one year.
No one can hear you scream.
That's true. The American Society for Parasitology sponsored a Science in the Movies session in which 3 of us gave talks, and mine was on Alien. I gave mine and all the flaws in Alien and how the parasitologists would cringe when they watched it. And they all agreed.
They also discussed the idea that virus-like particles, which don't have any DNA or RNA, have been found in ovarian tissue or the venom apparatus of parasitoid wasps. These have been well studied. And these can alter, all right, they call superparasitism behavior. Well, what would that be, superparasitism?
Parasitizing a parasite?
Yeah, I think so.
That's right.
So one of these virus-like particles, Leptopilina boulardii filamentous virus, alters superparasitism behavior and parasitism rates of the infected parasitoid wasp by increasing its tendency to lay supernumerary eggs in parasitized hosts. And
that means extra.
That's right.
We just talked about this in the pre-show, Alan, for 10
Get
minutes.
your supernumerary eggs here.
So you could have a supernumerary finger, right?
You could.
Some people have 6 fingers. That's supernumerary.
That's it.
But you can't have a supernumerary intelligence because that's not something you can count.
That's right.
You know, I don't hear that word used very often.
Supernumerary.
So that's an example of virus-like particles. Okay, so there aren't a lot of examples of RNA viruses in parasites.
In other words, Vincent, you're not trying to tell me that they produce eggs over and over and over again, are you?
Did you just think that up and said, I have to say this?
I did, because I didn't want to come on the next show and have to say it.
I'm glad you have a chance to exercise your humor. Were you ever a stand-up comedian?
No, I used to sit down a lot for that. No, I was called a stand-up parasitologist because I did inject some humor into my presentations, and I think that in medical school they deeply appreciated that.
He would paralyze the students and inject the humor into them.
Kathy and Alan, just one last
students.
question before we get back to this paper. Do you think Dickson has a sense of humor?
Oh, absolutely.
Yeah.
Oh, come on.
Some guy wrote a letter which he said not to read. And he said you seem not to have any sense of humor. Talking about me.
You have a
Oh.
great sense
He
of
says,
You do too.
I seem to be a very serious person.
humor. You all have a great sense of humor.
Well, that's okay. I don't mind what people... All right, so there are not a lot of RNA viruses in parasitoid wasps, so people haven't studied them very much. But now that you can sequence extensively, very quickly, relatively cheaply, but not for free, of course, people are finding more. And so here are some examples which have totally gone, over or under my radar? How does it go?
Under your radar.
Under. That's right. It's flying at 200 feet above the ground.
Yes.
So these have been in Nasonia vitripennis wasps, which are not rice, they're pastas.
That's right.
2 of these, which are Nasonia vitripennis viruses 1 and 2, are Iflaviridae in the order Picornavirales. So an order is when you take 2 families and stick them together. And the third virus is unclassified. The effects of these viruses on their hosts remain unknown.
But wait, on that unclassified one, it's called norovirus.
That's lovely.
Capital Nora.
It's named after
horrible. It's
Yes.
just horrible.
because it doesn't tell you anything.
Because it's going to be
With
With
What about
norovirus.
norovirus?
And
Norovirus. Is it norovirus or norovirus?
Yeah.
Well,
This one infects fruit flies.
It's probably named after the discoverer, right?
Yeah.
I do like Nora. It's a nice name. Then they have a new iflavirus named Dinocampus coccinelle paralysis virus, which has been found in wasps that parasitize lady beetles. And this is very cool. The replication of this virus in lady beetle cerebral ganglia. I didn't even know beetles had ganglia.
They do, of course.
Sure.
Most likely induces changes in lady beetle behavior. I want to do that paper, although after today's paper, we might not want to. But actually, this was mentioned by Nels Elde to me a couple of weeks ago. They heard about this at a meeting, and he thought it was pretty cool. So have a look at that paper. Tremors, gait disturbance, and limitations in movement. I don't know how you could tell that in a lady beetle. They seem to have tremors and gait disturbances all the time.
They're pretty smooth movers, actually.
So lady beetle. Is that the same as a ladybird beetle?
Yes. Ladybird beetle, ladybug, lady beetle.
They're used a lot in the biological control of aphids and things of that sort.
Yes.
I remember the wife of my first PhD advisor, Marie Hayashi, asking me, how do you know it's the female?
That's good.
They come in different colors, don't they?
They're wonderfully colored.
Red,
They're
orange.
absolutely beautiful.
Yeah, that's very pretty.
They look like little
was I gonna say
Volkswagens
about that?
with legs.
Yes. Right. So there are these other There are these a few RNA viruses that have been found in parasitoid wasps.
Mm-hmm. Yes.
Right.
Yeah, we just mentioned them, right?
Yep.
Now, non-segmented...
I was just catching up to try and...
We don't have all 7 reproductive strategies for viruses, though, do we?
Right. All right, now that they move on to mononegative virales, which are non-segmented negative sense single-stranded RNA viruses, like VSV, which we've talked about quite a bit lately. That's a mononegavirale. Mm-hmm. You know, and they filoviruses, paramyxos, rhabdoviruses. Most mononegaviruses have been found in vertebrates and plants. Only a few in invertebrates, and they list some of them with wonderful names. I like this one, soybean cyst nematode virus 1. Mm-hmm. So, is that a virus? It's found in soybean cyst nematodes. Now, what is a soybean cyst? A cyst of a soybean?
It's a disease caused by a nematode of soybean.
So where's the virus? In the nematode?
Inside the nematode.
Yeah.
Yeah, it's a nematode virus. Huh?
It's a virus it's a parasite within a parasite.
Is that a super parasite?
It's a supernumerary parasite.
No, it's not an extra one. These are related to tick-borne Naya viruses, NYA. In insects, rhabdoviral sigma viruses have been discovered. All right, here's a word that I don't know. Drosophilid. Is that Drosophila, basically?
Of course.
Yes.
Why? So
Drosophilid is pertaining to Drosophila.
So there are many different species of Drosophila. So
right. So a fly in the genus Drosophila would be a Drosophilid.
There you
go.
Just like a human is a hominid.
Yes.
And this is when these Drosophilids are infected with sigma viruses, they get paralyzed, or they die after you give them high concentrations of CO2. Don't regular flies die?
Anaerobes do.
But that's what you do when you're working on flies, to momentarily disable them so that they don't crawl off the microscope.
So the LD50 goes up.
Yeah. So here the uninfected flies recover.
Yeah.
That'd be something. You're putting out your Drosophila to do things, and then they never recover.
The other method is to bring them down to a low temperature so their flight wing muscles don't work.
I remember doing that in college with Drosophila. That was so much fun.
Yeah.
Only one negative-stranded mononegavirale has been found in parasitoid wasps. This is a very long name. Diacasmimorpha longicauda rhabdovirus. It must be.
Longicaudata.
That's the species, D. longicaudata. Right?
Right.
So that's basically why they wanted to look for some here, I guess. Or maybe that this was after they discovered it. They wrote
and they say that they don't have sequence available to confirm that it's really a rhabdovirus.
Right.
And its role in the wasp hasn't been clearly illuminated. So in this paper, they're going to claim to have identified the first bona fide negative strand RNA virus of a parasitoid wasp.
And what they say is this is the first report of an unambiguous mononegavirus.
Right. So
wouldn't the report be unambiguous, not the virus?
Yeah, I'd like.
No, it could be the virus.
The virus is unambiguous. Yeah. It's unambiguously a virus.
Yeah.
I want to try to interject some meaning to all of this, because what the system really represents is a control for cabbage butterflies.
Is that what these white, small whites are?
Cabbage butterflies. That's right. So they're larvae. The caterpillars eat the leaves of cabbage.
Is that a cruciferous vegetable?
No.
Yeah. Well, that's
cabbage?
That's what they say here.
Is it?
Yeah.
I don't know, Kathy.
Cabbage, cauliflower.
Cruciferous vegetable?
Cruciferous.
I didn't know that.
Yes.
Also collard greens and kale.
I take it back. It is a cruciferous.
Popular in Nolan.
And it's extremely economic to grow these in China, and they've got this problem.
Yes.
So they use these parasitoid wasps to control the butterflies.
Right.
So if they've got a skewed sex ratio here that a virus is affecting, so they don't get a lot of females, so they can't inject a lot of caterpillars, that's a problem.
Okay, we'll get to that.
We'll get to that.
The name of this wasp, this parasitoid wasp, is Pteromalus puparum.
Right.
The first name is spelled Ptero.
Yes.
So my question is that Pteromalus or Ptero?
No, Tero.
Because some people say apoptosis.
They do.
Yes.
So that would be Ptero if they're,
It's Tero.
Pteromalus puparum are predominantly pupal parasitoid wasps. Now, what does it mean to be predominantly pupal?
You inject their stuff into the pupa of whatever insect you're parasitizing.
Really?
Yeah.
That's what that means?
Why not?
Okay, fine. I wouldn't have put it that way.
Rather
Sounds
than
like
the caterpillar in the case of the butterfly.
No,
Because
but
they make a pupa also.
Never mind. They prey on butterflies of several species, including the small white, which is Pieris rapae.
Yeah, and that's a pest.
And over 90% parasitism in fields of cruciferous vegetables in China.
See that?
Here's another sentence. Puparum wasps are gregarious and synovigenic parasitoids. The Kathy knows what that means.
Synovigenic means they just keep laying eggs all through adulthood.
Wow. Right. And gregarious, is that what Dickson is?
They like
yes,
other
of course.
wasps of similar.
Yes.
Really?
breeding.
So a wasp can be gregarious?
Absolutely.
Wow. How would you tell?
Well, you can just find them all together.
Okay.
Hovering over a caterpillar.
Back on the wasp, the Pteromalus puparum,
Yeah.
I wanted to see what these look like, and so I pasted in several images from
you did.
a
This
Google
is
image
great.
search.
You did a great
They're
job.
these beautiful blue-green iridescent
They're gorgeous.
bodies.
They are gorgeous.
All right, I need more help here. The vitellogenesis of P. puparum is initiated after the pupal stage and completed at the adult stage after eclosion.
Right.
The vitellogenesis is when they start making yolk.
Oh,
Right.
and what's eclosion?
It's when they seal off so they can't be fertilized anymore.
Okay. Unlike parasitoid wasps with polydnavirus, this wasp has a different way to use venom to suppress the host immune system. So usually the parasitoids can use these polydnaviruses to suppress the host immune system, but these use venom.
And by the way, a lot of these, probably most of these parasitoid wasps don't sting if you're not if you're not their target species.
Yes.
Back on eclosion,
Yes.
I did my prelim exam as a graduate student on an insect question, and eclosion is the emergence of an adult insect from a pupal case, or an insect larva from an egg.
All right. I sit corrected.
Very good.
Yeah.
All right, so this lab studies Pteromalus puparum, and they were sequencing the genome when they stumbled upon a virus. Okay, and that's how science works.
Indeed.
What do we call that? Serendipity.
You got
Now,
it.
they were sequencing the genome or the transcriptome?
Transcriptome. Yeah, transcriptome. They're interested in the transcriptome.
You have to know when you've been lucky.
Which is the mRNAs produced by transcription of the DNA genome of the wasp.
And that's why they found an RNA virus.
They found a large contig, 12,141 base pairs. Everyone know what a contig is, Dickson?
Yes.
What is a contig?
It's a sequence which overlaps with another to produce a whole sequence.
Yeah, you put pieces together.
Contiguous. That's what the word derives from. Contiguous.
The contig was made many years ago.
Contig.
Maybe by Roger Stodden. Do you think, Kathy, that was his doing?
I don't know.
Anyway, I used a program years ago that would assemble a contig from small pieces of DNA.
I think contigs were first invented by sequencing the Caenorhabditis elegans genome.
I don't know about that.
No, I think that's when I first heard
why
that.
they thought this was a virus. This is very typical. It had regions similar to viral RNA-dependent RNA polymerases. That's often how you say, oh, this looks like a virus, because often the other proteins don't look like anything you know of. But polymerases have signatures.
Yes.
And so they sequence the rest of the genome. And it's 12,230 nucleotides with 5 large open reading frames. And there's no poly A at the 3' end. So if you knew just that, Dickson, what polarity do you think it would be?
Don't do this to me.
Oh, I'm just seeing how many different genome types?
7.
Okay.
It's reflexive.
Why is it reflexive?
Because by now I should know that it's always
Okay.
been 7.
Yes.
What else should we tell them about this? Let's see, 5' and 3' untranslated regions, so they could see transcription, initiation, and termination signals. There are intergenic motifs and termination signals. So this all sounds like VSV to me. Oh, that's a nice name. It all sounds like VSV.
All sounds like VSV.
And they confirmed transcription of the 5 open reading frames by doing northern blot analysis.
Yeah, that's
which unfortunately is relegated to the supplemental
figures, yes.
right.
And this is often the case, Kathy, that the first data are often in the supplemental figures.
Right. And not only that, but the method for it is in the main part of the paper, and the figure legend is in the main part of the paper. So you have to go 3 places if you want to look at that northern. And I still wasn't able to figure out. They just say it's total RNA. I want to know, do they grind up the entire wasp, or do they pull off the legs and the wings and then grind up the body?
Yeah,
but they didn't say.
Or the ovaries, which has the most anyway.
Right.
So these one of these open reading frames was shown. Its translation was shown to be similar to the Midway virus RNA-dependent RNA polymerase. And I just couldn't help but think if that was involved in the Pacific.
Right. It was a battle. It was a big,
there's a
very big
place,
battle.
Midway Islands, right?
That's right.
I wonder if they got it there.
The Battle of Midway was the turning point in the Second World War in the Pacific theater.
Yeah?
Yep.
Well, that's another podcast.
That's true.
Yes.
The other 4 ORFs had no similarity to any deposited protein sequence.
Yeah.
Exactly. Now, what does
amazing.
that mean?
It's very different.
It does. That's because they're interacting with with
an insect?
Well, not just with the insect, they're messing around with their reproductive strategies.
Well, VSV has 5, NPMGL, right? We talked about that in the classic Andy Ball paper, the 5 different mRNAs that can code the 5 different proteins. And I'll bet this virus encodes them, but they're different enough so there's no homology, right?
Right.
Mm-hmm.
But the only one you could see is the RNA polymerase, but they think that, there's an N and P protein, an M, and a glycoprotein as well.
Well, and as we said at the top, there are so few negative strand RNA viruses in these insects.
Yeah.
Not too surprising that this one doesn't look like anything because we haven't looked at others like it.
You remember a few months ago, a paper was published where they took 200 and some invertebrate species in China, and they just did total sequencing, and they discovered 1,000 new viruses. So maybe among those are some with homology to this virus, but wasn't in the database yet, and they didn't see it. Anyway, they concluded it's a mononegavirales on all this. And then they want to know, what is it related to? As we said, the RNA-dependent RNA polymerase is related to Midway virus. So they did some other phylogenetic analysis. And they notice when you compare to other mononegaviruses, this virus, which is called PPNSRV-1. The PP is the wasp species genus. And NSRV, what is NSR again?
Negative strand RNA virus.
Oh,
exactly.
Oh,
yeah.
I'm not sure about that.
7.
PPN, PPV.
They could have called it PPV1.
Yeah.
But, oh well.
Jens, what are you doing?
Well, maybe it was already named by the time Jens got involved.
But he's a persuasive fellow. So they feel this clusters as a distinct lineage in this family, Niamiviridae. And they think...
Right. I just wanted to say Miami sounds like Miami, but it begins with N as in negative. Yes.
There you go. It all fits. Good. It all fits.
It's very good. So they want to have a separate genus, a separate Niamiviral genus, and this will be a species in it, this PPNSRV-1.
Wouldn't this be a different family?
No, they call it a novel genus.
Oh, all right. Okay, but it's in the Niamiviridae.
In the Niamiviridae, right?
It's the family.
Yeah,
right.
you got SBCNV, SNVV, NY, Midway virus, those are all in there. And this one is different from those where they want to call it a different genus species. Okay. If Jens is on the paper, he must have agreed with it because he's the taxonomy person.
Yep.
And here is some comparisons. I only want to mention them because I love the virus names. Identity to the genome of soybean cyst nematode virus. We talked about that. Nyamanini.
Are you really?
Nyamanini virus. Midway virus. And Sierra Nevada virus. That one is made from IPA.
Yes, it has a lot of hops in it.
All right. So they say, all this indicates the need for a novel Niamiviral genus. I think it's funny. It's great stuff, but it's funny. Now, let's do some infection of wasps. So the funny thing is, the next section is titled PPNSRV-1. Tissue titers are highest in adult female wasps.
Right.
And they just say, viral titers in head, thorax, and abdomen were analyzed. How did you infect them? I would have said how we infected. They just pulled them out of nature.
They just started with colonies that were virus positive.
Are they breeding these in the laboratory?
Remember, it's vertically transmitted.
Or are they field collected?
Yeah. I don't know whether they're breeding them.
My guess is they're breeding the ones that home in on the white butterflies.
Okay, so they look in different pieces. The female body segments contain more viral RNA compared to male body segments.
Right.
And the viral RNA, only RNA, right? This is all qPCR. Virus viral titer in ovaries and digestive tract and body remnants. Testes had the lowest viral titer. And so maybe it's not transmitted via testes, right? Well, not testes, but
by sperm.
By sperm, yeah. It's detect viral RNA in eggs, larvae, pupae, and adults.
Right.
And during development from larval to adult stages, the viral titer increases. Of course, the RNA increases and peaks in the adults. And the genome copy was highest 4 days post-eclosion. What a great name. Word. What else is interesting here? Okay, that's enough of the distribution. So they've looked at that. Anything else, Kathy?
Well, I can tell you a little more. Insect rearing is the first method they discuss.
Okay.
They initially collected them from cabbage fields in China and maintained as described previously. But they took offspring from a negative breeding pair or positive breeding pair to produce the positive and negative colonies that they're going to use later.
Okay. All right, then they did a little immunohistochemistry.
Yeah, I saw that. That was nice.
They take wasps, they embed them in paraffin, fix them, they slice them, stain them with antibodies. So they find viral antigen. So I suppose they made an antibody.
Right.
Which must be described in the methods.
Yes, it is.
Is that a virus preparation or is it a protein that they produced? Do you know?
cDNA fragment was amplified. PCR products were cloned, expressed in E. coli.
Okay.
Recombinant protein. His-tagged.
So they see antigen in ovaries, eggs, midgut, testes, and seminal vesicles. And they have some beautiful images here.
Yes, they do.
Lovely.
Really are lovely.
They do.
You can see the insect parts and the lovely staining and dimensionality. And also in the leg, I think. Is that a leg? I don't know what that is, actually. There are little fibers of green in C1 running. Do you have the paper, Dickson?
I do have the paper.
You see panel C1 that's blue with little fibers.
I'm about to go to
green
the paper right
around
now.
And perpendicular. It's very pretty, which, of course, is a secondary thing here. Is D1 an ovary? That could be, huh? Let's see. D1.
Some of these could qualify as entrants into the Small World contest that Nikon sponsors.
D says it's sperm.
Yeah, just say sperm.
Sperm, right.
I don't know the difference between sperm and ovaries.
Well, you would if you were a female wasp.
So they see antigen in all these tissues. In the ovarioles, the signals are higher in oocytes, follicle cells, in the intracellular space compared to nurse cells. No antigen in tissues from uninfected wasps. So I guess they screen wasps, and they see some that are uninfected, and they can section those and see that there's no antigen in them. Right? So if you go out in the field, you can find wasps that are not infected, right? So it's not 100% infection.
Exactly. That's true.
They prepared sections of digestive tract and ovaries, did some electron microscopy.
Yes.
And they can see virus particles.
That's correct.
Yep.
By transmission, electron microscopy, 60 to 100 nanometers in diameter.
Right.
In ovaries, follicular cells, and cells of the digestive tract. And they say the spherical particle is similar to that produced by Naya viruses. Now remember, this is not like VSV, which is bullet-shaped, right? These are spherical. Of course, other mononegavirales are spherical, like the paramyxoviruses. Dickson, are filoviruses mononegavirales?
Yes, they are. In fact,
are they spherical?
No, they're...
Yeah, there's a picture on my wall. Very good.
Ebola.
Filamentous.
Thank you.
All right.
Philo dough. They also
virus.
infect Greek pastry.
Now, Naya viruses, remember, are in the Nayaviridae. And they want to put these new viruses, these PPNs, into the TWiVumvirate family as a separate genus. Okay, next, they show vertical transmission by the wasp. Vertical transmission from both females and males to offspring, although the patterns are different. It's very interesting. They have a picture here, and I don't know why they have to have pictures of wasps on this figure.
Because they're pretty.
Yeah, they
Although
got
the pictures are kind of small.
they are very small.
There.
So infected, when both are infected, the male and the female, you get 100% transmission. If they're uninfected, surprise, you get 0% transmission.
That's reassuring.
If the female is mated with a uninfected male, all of the offspring are infected, both the males and the females. However, if the female is uninfected and the male is infected, you only have transmission to females and only 58% of them. It's interesting.
Mm-hmm.
But it is transmitted vertically. So all you have to do is mate and check the offspring.
Right.
And you see the virus in them as well.
Mm-hmm.
Straightforward. Mm-hmm. Now they stick in here a paragraph on the geographic distribution. I think they should have followed this up with the offspring sex ratio, but they didn't. I don't know why. They collected wasps from 5 different locations in China to see where it is, and basically the virus was found in 4 wasp populations, all in the Zhejiang province.
Yeah, the higher up they went, the more they found.
Yeah, the prevalence decreased with dropping latitude.
Exactly.
So dropping latitude is
not altitude. It was latitude.
No, I know it's
latitude.
But
closer to the equator.
Yeah, exactly.
So if you go north,
yeah,
and it gets colder,
yeah, apparently,
right.
So not all of China is hot.
Oh,
right.
my no.
Let me look at the graph here.
My goodness.
The map. So we have Hefei, Hangzhou. Where is Beijing here?
Northeastern. It's almost in Manchuria.
Okay. So as you move north, you find less. But the positive ranged from 16 to 37%.
I mean, I would like to see the distribution of cabbage patch.
Well, it's interesting, too, because there's no correlation with temperature or latitude in October.
Mm-hmm. Right. Yes.
Which is after the harvest.
Right.
So I think this is an agricultural problem. That's what I think.
The virus could not be detected in unparasitized butterfly pupae collected from all 5 locations. You would expect that, right? No wasps, no virus, right?
That's right.
Now the harvest would occur at different times in different regions, though.
That's all true.
Right.
And the other wonder that I have now that we're talking about the caterpillar, and wonder what effect any of these virus proteins would have on the caterpillar, on the butterfly itself.
Right.
Now, not the wasp, the caterpillar.
Mm-hmm. Right. Yeah, we didn't look at that, right?
Nope.
Sorry.
They look at the parasitism rate.
Yes.
Is that not what you were talking about, or no?
No, I want to know if the virus can infect the caterpillar as well as the wasp larva.
Mm-hmm.
Got it. Okay.
Yeah, we know the RNA is there because it's in the wasp eggs, right?
Exactly right.
Right.
And you could express each protein and then see whether it had an effect on the biology or not.
Wouldn't you want to take sections of the white butterflies, and then see if you have protein produced?
Absolutely.
All right. Comparison of the positive and negative virus positive and negative wasp colonies. No difference in successful parasitism rate and total oocyte number.
Right.
But offspring sex ratio was changed. Was lower in the plus colony. So female offspring number emerging per parasitized pupa, the offspring number laid by female wasp, and adult emergence were lower in the infected colony compared to the uninfected. And the longevity of female and male wasps was longer than compared to uninfected.
That's fascinating.
Longevity?
Yeah. I mean, here you have it extends the life of the parasitoid wasp, but it skews the sex ratio in more males than females. Right. So how does that work out in terms of a positive or a negative effect on the evolution of this relationship?
How much time are we talking about here? Does anyone have the number? Like when we talk about longevity, how many days longer?
Oh, I see what you mean.
I don't know what these numbers mean. They have T values here, and I don't know what they mean. Figure 5F. Let's take a look.
Got to be in
here we
these.
go. Successful parasitism ratio, offspring. Oh, here, adult longevity in days on the y-axis. So uninfected, males 34 days. Males 15 days. Females, I would say 18 days. And then infected males 18 days and females 25 days. So females go from 18 to 25 days. It's a week.
But in the life of
a lot of
insect, that's a big percentage of its lifespan, right?
What is the lifespan here? Do you know?
You just said it.
18 days,
I did just
25 days.
I did just say it, yeah.
Yes.
So that means that there's a 20% or 50% more chance that they would infect a host with their eggs if they lived longer, but there are fewer females to do that.
Right.
Mm-hmm.
But I wonder how the females actually find the caterpillars, because there's some pheromonal stuff involved in all this too somewhere.
Odors, right? Chemicals.
Yeah, absolutely.
Yeah.
And this is really strange that it would skew the sex ratio in that direction, considering that it transmits better when it's in the females, right?
Right.
Mm-hmm. It is weird.
There's some weird evolutionary biology going on here, or more likely selective pressures that we don't understand yet.
Oh, for sure.
Right.
Yeah.
For sure. At high temperatures, longevity was still longer compared to uninfected.
Right.
And female offspring laid by each wasp per day for the infected colony was less at higher temperatures.
Yeah.
Okay. Now, these are all done by looking at already infected wasps. So they wanted to know if they actually injected the virus in, would that make any difference? So they inject virus into yellow pupae from negative wasps. Now, what's a yellow pupae? Does anyone know?
It's a younger than older wasp.
In the one figure, they show white pupae and black pupae in different developmental stages, but they never show us yellow, so it's hard to know.
Is that dimorphic maybe for males and females?
I don't think so.
No.
Just yellow pupae.
So these are wasp pupae, right?
Right.
So then you let them mature, and then you see how they reproduce. That's the experiment, right? So you don't inject it into an adult wasp.
No.
Okay.
Right.
And
what they're using to inject with, this is some kind of, I think, semi-purified virus liquid.
Which is from infected wasps?
Right. Mm-hmm. So to do the EM, they tell us how many thousands they had to grind up, and then this was a different kind of a prep.
Oh, my goodness.
To do this.
This sounds like it would grow in insect tissue culture very nicely, though, wouldn't it?
Well, and yeah, that was one thing that I don't, well, now's the time to talk about it. They do all of their assays for virus based on the RNA. Mm-hmm. And if they had this semi-pure liquid, and if they had insect culture,
plaque,
do plaque assay. And maybe they tried it and weren't successful, but then that would be interesting to know.
Sure.
Yes.
Yeah.
So maybe they did, but maybe they didn't.
Negative results are important.
Very.
Yeah.
Because then other people, we're not going to try this. Well, on the other hand, sometimes the other people can get it to work, right?
You bet.
They still would like to know if they tried it. Anyway, basically, when they inject virus, they get the same results in terms of parasitism rate, sex ratio, offspring number, etc. There's no difference. So what they observed was correct. Right. All right, what do we have here? They injected double-stranded... Oh, I see. So you can... Now they wanted to knock down one of the viral ORFs, ORF1. And in insects, you can inject short double-stranded RNAs, and it will very effectively knock down any target by RNA interference. In cell culture, in insect cell culture, you could just throw the RNAs onto the cells. It's very, very efficient.
There you go.
Do genome screens that way. It's really cool. So they put a double-strand RNA targeting ORF1 into yellow pupae from infected wasps to see what would happen. And so it knocked down the ORF level, as you would expect. And no difference in parasitism rate when they did this. Am I reading that correctly? A lot of numbers.
That's right, paragraph. Yeah. If you just look at the figure, this is open access, by the way, because it's SPLOS pathogens. So it's Yeah, there's no change in the parasitism rate. Maybe a slight change in the offspring sex ratio. Offspring per parasitized pupae. They don't do the best job of putting all of this RNAi data together for you in a concise sentence or two.
They only looked at,
one of the ongoing annoyances I had with this paper is the way they split the figure legend. It's minor if you're scrolling past it on the screen, but every figure, the legend is either on the next page or it's split partly on the next page, and it's just a little.
Yeah. Just make the figure a little bit smaller and
just a
put
tiny
the legend
trinket to the figure, and you could put the whole legend on it. And I have no idea why it was done that way.
It makes it a lot easier to read them, obviously, instead of have to scroll back and forth.
Yeah. So Senator Eisen, that's better get on that.
But they only targeted one ORF, correct? By this, they should have done all of them. They should have done all of them.
Maybe they're working on that.
So what is ORF1? It's probably the first ORF in the genome, right?
Right.
And
it's not the RDRP one. I looked at that.
So that's probably what, NP?
Yeah, something like that. N or NP, whichever. I can't remember. Yeah.
Yeah, N is first, yeah. So that's the protein that coats the genome. I'm surprised that knocking that down has no effect. But maybe at this point, the virus is replicated extensively and its products are there. And but you're doing an offspring study after knocking down, so it should have an effect. So I don't understand. Do you understand why it had no effect, Kathy? Knocking down ORF1? They don't really explain it either.
Right.
So weird. That's very strange.
Also weird that they didn't do the other four just to show that they'd followed through for the entire genome.
If none of them had an effect, I would be worried.
Exactly.
Right.
That there was some problem.
Yes.
So so the genome copies with the RNA injected wasps declined compared to the control in both sexes.
Right.
Right. So that did happen.
That makes sense. But the parasitism doesn't change.
Parasitism
the sex ratio,
rate didn't change. Sex ratio.
No, it doesn't change. The control, it went down a little bit. The GFP control.
Well, yeah. Well, no, they do say all these things, sex ratio and so forth, when you get to page 13, say that they were significantly higher in the RNA-treated colonies compared to the control colony.
Yeah, that's true. Significantly higher. So that's taking away the effect of infection, basically, right?
Right.
Yeah.
Right.
All right, so there is an effect. Sorry about that, folks. Right. So there you have it, a new negative strand mononegavirales of parasitoid wasps seems to have. Now, Kathy, what does this mean? It mediates the secondary sex ratio.
Well, we had this discussion, and we talked about the fact that we know what secondary sex traits are, but I think the word secondary is superfluous here.
It's supernumerary.
Yeah, because I don't know, what's the primary sex ratio. So maybe there's an entomologist that can inform us what that means, but we weren't able to come up with that.
Right.
I'd like to know more about this system too, in terms of how the wasp finds the the victim.
Yeah,
and then what happens afterwards? How does it actually end up killing the caterpillar? Just eats it alive, or?
Are you interested in whether the virus has any effect on the wasp?
I would like on the wasp. Well, we already know that, but I would like to know if it has an effect on the butterfly.
It has no pathologies, they say.
Pathology is one thing, but maybe it alters.
It has effects, but they're saying they consider it a non-pathogenic commensal virus of the wasp. It has no pathogenic.
No, I got that.
Okay, good.
But what effect does the virus have on the
butterfly?
On the butterfly?
Yeah, that's what I want to know, if any.
They did not address that here, but they could.
Next paper.
They could. And they have, by the way, submitted this to the ICTV, and they have accepted their proposal of having a separate genus with a single species. They're going to call it Pteromalus puparum paropuvirus.
Just rolls off your tongue.
So that's going to, Kathy, we're going to get rid of PPNSRV-1.
Okay. I'll be happy with that.
So this is the first mononegavirales of insects. Yes, the first first Niami virus that infects insects. The others do not infect insects. Vertically transmitted and distorting the transmission ratio as well, depending on who's infected. Males transmit exclusively to females, which is very interesting.
Indeed.
Very different from Wolbachia.
There you go.
Right.
Yep.
So they say
Go ahead.
They say the virus, this is cool because the virus is transmitted within sperm or attached to it because in haplodiploid species, the sperm-fertilized eggs develop into females while unfertilized eggs develop into male progeny. So that makes sense now.
Parthenogenesis.
Right?
Yeah.
So the males that are infected, if the sperm get to infect and fertilized eggs, not infect eggs, fertilized eggs, they develop into females.
Mm-hmm.
And that's why the
Right.
females are infected.
Right.
And the eggs, unfertilized eggs, become males, whether they're infected or not. So the virus would go into males that way. So that is what their explanation is for this funny transmission ratio.
The parasitoid wasps have weird sexual deviations from normal biology. They really do, because they can also reproduce parthenogenically, and
Some of them can, yes.
some of them can. And the Wolbachia aid and abet that process, as I recall.
That's
They're a
interesting.
perverse genus.
They are perverse.
And remember, this genome is... Wait, they don't know this. They know this is in the transcripts. Did they look in the genome of the wasp to see if it's integrated?
Good
No,
question.
they didn't. Because
Very
they
good
say here,
question.
maybe the genome is integrated and transmitted vertically through chromosomes, but they didn't look at that. You would think that'd be very easy to do.
It is very
right?
easy to do. Yeah, sure.
Hmm. And, okay, so that's basically it. How the wasps are infected, they don't know. That's interesting, right? I mean, they're not 100% in the fields. So you can ask how they get infected. Besides transmitting to the offspring, do they get infected de novo, right?
Mm-hmm.
Is that a reasonable question?
It's very reasonable.
And they would like to know that what is the underlying ratio of this secondary sex ratio?
Yeah, yeah. I mean, if you had an uninfected female that injected a caterpillar with an uninfected larva, is that as effective in controlling the outbreak of butterflies as the other?
Right. So they note that in arthropods, many vertically transmitted microorganisms can alter sex ratio.
Yes.
Including Wolbachia, the best studied,
See?
right? Which
That's
we
right.
have talked about.
That's right.
They do a lot of things to...
Yeah.
They do feminization, parthenogenesis, male killing, and sperm-egg incompatibility. Cytoplasmic incompatibility, that's what we talked about. It's due to the lysogens
That's right.
Right.
in Wolbachia. That's very cool.
Yeah.
And then there's a masculining virus in the common pill bug. That
Sounds like Rich Kessin should know
a
about
masculinizing virus and a male-killing virus like RNA sequence in the Oriental T. tortrix moth.
that. Sounds economically important.
They say that their virus to meet the pattern is consistent with a female-killing phenotype, so it's another way that it might be doing this. Although male-killing sex ratio distorters, you can't make up this stuff.
No.
Male-killing sex ratio distorters are common in nature. Female-killing is rare. And that makes sense, right?
Right.
Well, there would be a selection for that. That's true.
And that they say it would seem to be maladaptive for the virus as it's transmitted vertically through females, yes. But this is different because it's also transmitted through males, which is quite unusual, they say. So very interesting. There are lots of interesting questions here. I love this behavioral change thing. I think it's great, and I like the sex bias change, or whatever we call it. Sex ratio distorting elements, SRDE. And the longevity is another thing. Makes you live a week longer. I think we should get this virus for people and make them live longer. Would you like
them
it,
live
Dickson?
a week longer?
It depends on if I knew how much I had to live in the future. Whether I
Well,
would
Alan,
accept the virus.
a week is like 50% of the 25%. So
It's a lot of time there.
you can get an extra 25 years, Dickson. You could be 100 with this virus.
Yeah, I could. I don't know.
Can a virus make you live longer? Just like we had can a virus make you fat?
Let's just.
Now it makes you live longer. Oh, cool. So that's it. Anything else we forgot?
I did find that you were right about Staden coining the term
Yeah. Roger
Contig.
Staden.
Saw that.
and also The Midway virus was first isolated in 1966 from seabird ticks of 2 species collected on the Midway, Kure, and Manana Islands in the Central Pacific and from northern Honshu.
Yeah.
Look at this. Ticks of seabirds.
Yeah.
I mean...
Successful systems attract parasites.
No, but who's going to be looking in ticks of seabirds?
Tons of people.
For viruses.
Are you kidding? Are you
Yeah, tons of people?
Tons. Ticks transmit diseases. So
I've never met any of them.
By the way, the tick was the first arthropod shown to transmit a disease from one organism to another. Theobald Smith showed that.
There's a hall named after him.
Along with
Rockefeller.
Kilbourne. Yeah, I was in that hall for over 3 years.
Yeah, Smith Hall, yeah. I think
But
that
Theobald Smith,
was Cross in that hall?
yes, I believe he was. So was Peyton Rouse.
And Traeger was in there too, right?
Yep, Peyton Rouse was there. Shope was there.
I've been there. It's a hallowed ground.
It is.
This episode is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting high standards for ingredients, and building a community of home chefs. What they do is deliver seasonal, not seasonable. It's a big difference. Recipes along with fresh, high-quality ingredients so you can make delicious home-cooked meals. Every box comes with a step-by-step, easy-to-follow recipe card and all the ingredients pre-portioned that you need, except salt, pepper, and oil. But most people have that, right, Dickson?
Right.
And if you spend a lot of money eating out or at high-end grocery chains, this will save you money, less than $10 per person. And you're making a home-cooked meal, plus you're continuing your science at home. You know, you mix stuff in the lab and you go home, you mix stuff, except here you can eat it, whereas in the lab you can't. How do they do this? How do they make it so cheaply?
Reasonably, please. It's not cheap, it's reasonable.
What would you say, cheap? $1?
No, cheap is like that's a derogatory term, I think. But,
inexpensive.
Right. Affordable.
If these were $1 each and excellent, that would be amazing.
Oh my!
But that's years ago. That's years ago. Yeah, these are affordable, I would say. Although, if you're a graduate student, it might not be affordable to spend $10.
That would put Blue Apron in the red, by the way.
As a student, I ate at a diner around the corner from Mount Sinai for a couple bucks a meal.
I did. I ate at our diner over
Whiskey
here.
down.
Oh, really?
I used to have whiskey down. That means toasted rye bread. Because whiskey, rye, get it? I went to a Greek diner. You say, "Oh, boss, you want whiskey down?" Yeah. Those great names they had for stuff. Whiskey down. Okay, back to Blue Apron. They have a network of farms, fisheries, and ranchers that they contract from. And they say, give us your stuff, we'll give you money. Fish, animals, plants, and all done responsibly. They don't overfish. They don't treat their animals poorly. Responsibly raised animals. They do regenerative farming for vegetables. And they just give you what you need so they don't waste food. You don't overeat. You can customize your recipes every other week, whatever you want. You can get meat, non-meat. They deliver to 99% of the U.S., and they don't repeat their recipes, so you're not going to get the same thing in 1 year period. And this month, they have salmon piccata with orzo and broccoli, which started our orzo. Let's check out...
I have some of the ones for this week.
Yeah, tell me.
This coming week. It's still in time to order. Kale and white cheddar quesadillas with radishes and fried eggs.
Nice.
Seared chicken and French lentils with arugula and feta salad. Southern Italian cod stew with pizza bianca.
Wow.
Yeah, and here's one with crucifers. Creamy red rice and cauliflower casserole with spinach and rosemary.
Cool.
Nice.
They have a mixed mushroom bucatini. You know what bucatini is? It's not rice.
I don't know what it is.
It's pasta. The fat. It's like spaghetti, but it's fatter.
Oh,
I have a problem with sorry.
Is it shorter than spaghetti?
Yeah, it's a little shorter, but I have a problem because it's so thick that when you twirl it on the fork, it sprays all over my shirt.
Oh yeah,
real problem.
Yeah.
So I end up doing the thing that you should never do with spaghetti: cutting it. Never cut it.
Goodness.
So I don't eat bucatini because of that. I only eat spaghetti or angel hair pasta because you could twirl it around.
Put a red shirt on.
Does everyone twirl their pasta around their fork?
I do.
Yeah, okay.
I use a spoon, but I mean, I pour into a spoon.
I grew up, I was taught that.
You know what
growing
helps?
up, and then I went to Italy and the guy said, no, you should not use a spoon. You should put it right into your dish.
Throw a lot of cheese on it and it'll stick together and it won't spray so much.
I love grated Parmesan.
That's right.
Check out this week's menu. Get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiV. That's blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron is a better way to cook. By the way, Alan.
Yes.
You heard on Twitter that you can actually find the recipes online?
You can find the recipes, yes. One of our Twitter followers, I forgot his real name, but it's Colm on Twitter, pointed us to their... Who was that?
No, it's just, their recipes have always been online.
Right.
That's all.
We didn't know.
Yeah.
You didn't know. Okay.
I didn't realize they were all organized on one page. You just go to go to the blueapron.com/cookbook and you get this whole big, and you can sort by ingredient or cuisine or season and get the whole thing.
_Colm.
Yes.
You know what that means, Dickson? _Colm. You know what an underscore is?
Yes.
So a lot of people have Twitter handles with underscores at the beginning because that way they can get the name they want, otherwise it's taken.
Oh.
And people pronounce it _Dickson, for example. So you could get _Despommier, even though Despommier is taken.
No,
not that you would want it.
If Trump tweets, I don't.
But that's okay. There are 2 fake Dickson Despommiers.
It's all fake news anyway.
You know, that's an honor. There are 2 people imitating you.
Yes.
They think so highly of you. They love your meme, and you don't even get social media.
You're right.
And they do a good job, by
Good
the
for
way.
them.
I thought one of them was you. And,
well, I'm proud of me.
Yeah.
Hannah writes, Dear esteemed TWiVumvirate, I'm Hannah from the northern suburbs of Boston, where as usual for February the days are getting longer, but the winter weather just won't let up. It's 36 F2C with patchy clouds now, but we're supposed to get a snowstorm tomorrow. Yuck. I don't know if I'm too late to win the book since I listened to the episodes a week or 2 after they air, but I thought it would be a good excuse to write. I studied computational biology briefly before switching to a career in the software industry. However, I still love to learn about various biology topics, so I'm a big fan of all the TWi series podcasts. They're fun and convenient way to learn about new research while driving, cooking, and doing monotonous chores. Unlike most other science podcasts, which just give quick overviews of studies, you really get into the details and explain them in a clear, understandable way. I also enjoy Alan's puns. Here's one of my own. What do you call an asymptomatic stomach virus? Ignorovirus. Thanks for taking the time out of your busy schedules and research to produce these great podcasts. Keep on twiving, Hannah. And she has a listener pick, which we'll save till later. All right. Alan.
Stig writes, Hello, Twivsters. First-time emailer, and I guess you could say long-time listener. I would like to throw my hat in the ring for a chance to win the book Infections of Leisure by David Schlossberg. The temperature here in Denmark is unfortunately continuously below 0°C this month, in the mornings anyway. My name is Stig. I'm an MSc student at the University of Copenhagen. My BCS is in biology, and I will soon have my MSc in microbiology, which I will have to hand in at the end of August, although my master's thesis project is in bioinformatics, or as you would say, computational biology. I was fortunate enough to be able to do my master's thesis project at the local hospital, where I have the support of the other much more experienced bioinformaticians or computational biologists. And then I guess this was a note to self in the email, something about thinking about taking a PhD in computational biology might not make it into the final email. Oh, I guess we weren't supposed to read that. Okay. Continuing, Infections of Leisure would be a nice addition to the office where I have been given a desk. Right now, all that I have there is a dozen printed underlined articles and lots of papers from the union that I am now part of, and of course the Linux machine from which I work. And IDA releases one newspaper a week. I'm not sure what that is. I've been listening to TWiV, TWIM, and the newer podcast TWiVo for under a year, and unfortunately, it's hard to find time to listen to the back catalog, as we have a child, our lovely son Sixton, who is now almost 20 months old, a part-time job, and my master's thesis to attend to, although I would like to. I usually listen to the podcasts when I'm at work, student job at the LAF bench. Having been working in the same place there for almost the entirety of my time at the university, coming up on 5 years this October, it's easy to let the hand do the work when I listen to you discussing the interesting topics. I would also like to thank you all, the hosts, and all others involved in making and supporting through Patreon or Cafe Press, the Twix family of podcasts, the hours of entertainment we can enjoy for free because of all the hard work you put in. Lastly, I would like to thank the hosts for the important task of commentating science to the public, a task not easily undertaken, but you do an amazing job. One more thing, even though I'm studying to become a microbiologist, I find TWiV to be the most interesting of the podcasts, mainly because you usually also cover other important topics than the papers you present. Keep up the interesting conversations. So, studying to be a microbiologist, well, TWiV is microbiology. Virology is part of that.
Indeed.
Yeah, I agree. But I've asked Elio Schechter, are viruses microbes? And he said, no.
Of course not.
Well, I guess...
What does he think they are?
Viruses. But they're studied in...
I guess we do
they're
have,
mini
have the
microbes.
ASC and the ASM. So.
Well, that's a political thing.
I know
that's different.
Yeah, that's got
when you get questionnaires as to what your discipline is,
Right.
they almost never list virology, and you pick microbiology.
Yeah, I do.
Viruses or microbes?
And
I would have known
parasitology,
when
too.
Elio was here, I would have
I get
told
the
him.
same
Yeah.
deal.
I agree.
At the other end of the spectrum, I should say.
I have a degree. I have degrees on my wall that say microbiology.
Damn
so.
right.
Yeah.
I can't argue with Elio because I have such respect for my elders.
You do?
That's a joke.
You do? All right.
I do, Dickson.
All right,
It's
all
just
right.
that you burn me up.
I guess so.
Dixon, can you take the next one? You could take the next two.
I'll take the next two. I wish I could have taken the third one, but
That's going to Kathy.
I guess.
I look
Okay.
ahead.
I read them all already. Temet, right. That's because I rehearse. I assumed 27 would be reached quite quickly, but I guess not. I decided to pull the vehicle over and try my luck.
Didn't they tell people to pull over?
You did.
We did. Yeah, that's great.
You did. Thanks for everything, Temet. Richard writes, Dear Virus Mavens, I would be grateful if you would do a TWiV in a lab. Tell us what the lab looks like. What are the sounds and smells? How big is it? Who is there? Are they sitting or standing? If they use an apparatus, what does it look like? How does it work? When you need a reagent or cell line, how does it come? What does the container look like? Who does the housekeeping? Do they have to know what not and what to touch? You bet. Are there clerical staff associated with the lab? If so, what are their names? Have they ever saved the day? I might understand the science a little better if I understood the lab better. And Kathy writes,
I think that's a good idea.
I think it's a fabulous idea.
In my experience, the answer to who does the housekeeping is nobody.
Nobody does here anyway.
That's a very...
So let's think about this
you
a bit.
mess it, you clean it.
We should find a lab, not my lab because there's hardly anyone in it. So nothing would be happening. But I'm willing to pay for TWiV to go to one of our labs to. Kathy, do you have a busy lab? Would things be happening? We could record right in the lab.
Yeah, maybe not so much, but I think it'd be fun to find yet another lab that isn't one of ours.
We have to find one of our friends.
Yeah.
Who would be willing to let us sit in the middle with all the hubbub and record while people are still working, so we could say, "Oh, look what they're doing. They just broke a beaker."
We
Someone
might
just
be able,
injected themselves with Ebola.
we might be able to combine this. We're all going to be in Madison for ASV, right?
We are.
We are, yeah.
And there are certainly labs at Madison.
There are.
Mm-hmm.
Yeah, I think that might be technically hard to do because of the location of the TWiV compared to that.
There's
but
a lot of
we can keep thinking about it.
And
Maybe sometime when we're already going to go someplace, Vincent, like when you're going someplace. I mean, it really should, I think, have a camera though.
Yeah, we'll have a camera for sure. It would be good to have a crew so they could turn the camera onto someone in the lab, right?
Yeah, but they could just watch CSI Miami.
No, this is TWiV. Dude, this is TWiV.
To answer one of Richard's questions, who does the housekeeping? Do they have to know what not to touch? Basically, the housekeeping would be your typical custodial staff who will empty the wastebaskets and sometimes sweep the floors. They never touch anything on your bench or anything anywhere else. Right. So they just don't touch anything. And they all pretty much wear gloves, even when they're just pulling trash.
Yep.
Yeah.
So. Yep. And they don't sweep the floor. You have to pay them to sweep the floor, especially. Yeah, they just empty the trash pretty much. Yeah, we'll do that. I think that would be a lot of fun. Keep it in mind.
This reminded me of someone asked a long time ago, what's a day in the life of a virologist like?
Yeah.
And so Vincent and I did that on TWiV 172.
Yep.
It's not the same as what's it like in the lab, but you might enjoy hearing that if you haven't.
Yeah, in the meantime, but we'll do that. We'll figure something out. All right, Kathy, you can take the next one.
Lord Klingon writes, emergent infections will come out to play if you eat just one rotten apple a day. They can make a placebo just go away. The power of suggestion has nothing to say. When you get pricked by a needle with a roll in the hay, then start to feel ill, here's what you say. It's only emergent, it hardly sounds urgent. I'll wait till it's rampant or mildly insurgent. Then pick off a scab, send it down to the lab. We found something small and it looks like a crab. Here's what to do, it's all up to you. Go down to the beach and swim out of reach of the lifeguards and coast guards and people who screech. He's got crabs. If the power of the brine can cure a cut knee, if that doesn't work, then drink your own pee, or smear on some yogurt, or drink herbal tea, an old mother's cure, or home remedy, because we're all busy doctors, so don't bother me. And then Lord Klingon writes, please don't send a book if I win. I can't be bothered reading them. Or as Marie Curie once said, no good can come from reading books. Get out there and do something. Maybe I mistranslate the French a little?
A little.
Does this continue
yeah,
here?
It's all him. Yeah.
Okay. I've made a guess as to why you chose Omega Tau to be in your podcast stable and thought of Marcus Volter's own description of his podcast. Conversations with people who know things.
Yeah, that's pretty good. I like that.
Mm-hmm. I heartily recommend the Amp Hour podcast. Two lively gents, excellent in-depth content, wonderful guests with candid interviews from the cutting edge. They are all letting off steam and sharing delight, much like yourselves. In a recent episode, I liked Dave Jones' prosaic Australian outlook on people who are anti-science. Quote, don't bother pandering to their anxieties. Just relentlessly shame them for being stupid. End quote. He, he, he. Okay. Keep on yapping, you lot. Many thanks for the hours of listening. I can't read books no more through an inability to...
I think that's great.
There. Yeah.
DONC. I like the poem. It's good. Yeah. You like it, Dickson?
I love it.
Good. Let's do one more round.
Mm-hmm.
All right. John writes, hi, TWiVumvirate. Thanks for the continuing podcasts. I finally started Dr. Racaniello's virology course, and it is great to finally have a better understanding of some of the jargon in TWiV. Unfortunately, my lack of biochemistry, biology, and chemistry background is beginning to hurt my progress. I might have to dive into the rabbit hole of learning the prerequisites for the course. Oh, well, who needs free time anyway? Thanks again, John from Limerick, Ireland. Dickson, can you say Limerick in an Irish accent?
From Limerick.
Very nice. All right, Alan.
That's not a good accent.
Sure. Okay. Vasudev writes, hi, Vincent et al. Long-time listener, but I rarely get an opportunity to write, as I almost always listen to the Twix podcasts during my arduous commute on the Beltway. Today, I got the opportunity to pen a long-due email as I'm listening to TWiV on my flight to CROI, the Conference on Retroviral...
It's right below. Retroviruses and opportunistic
Retroviruses and opportunistic infections. Thank you, Kathy. Also, some of the previous TWiV guests whom I don't want to name due to privacy concerns are on this same flight, which makes listening to TWiV even more exciting. Cool. As a neurovirologist and as a program officer with a neuropathogenesis portfolio, I was so excited to hear an episode on neurotropic viruses and to listen about the blood-brain barrier, cytokines, and TEER something I worked on quite...
It's TEER.
TEER.
Something I worked on quite a bit in my previous life. Transendothelial.
Oh, yes. Right. Something I worked on quite a bit in my previous life. I'm really screwing up the abbreviations today. I was surprised to hear that you haven't yet gotten... Yes, tear pressure. You better call the BBB and complain.
Indeed.
You haven't yet gotten to the 27th email, and also wanted to write in to applaud you all for your never-ending commitment to science communication. Also wanted to get in touch with you about your idea about a museum with 3D models for infectious agents. I guess this was discussed on the podcast with Nels. This is a long-term dream of mine, as I know firsthand from NIH science fairs that lots of kids are absolutely fascinated with 3D models. If you're really serious about this project, I can volunteer my time to work with the Smithsonian and NIH, NSF, ASM to make this a reality. By the way, I was one of the first listeners to write to you about Zika way back in 2011 or so when it was not a big news story. Keep up the good works, folks. Would love to meet and chat with you folks in person sometime. Vasu. Big fan of Dr. Rich Condit. Not that I'm any less appreciative of the others. And Vasu is at NIMH.
I think it's funny that he has privacy concerns of previous guests. Well, if they're on TWiV, they're not private,
They're not
right?
private.
Yes, I'm very serious. So I had an idea, which I talked about with Nels on a TWiVo out at Utah. I said there should, because we did that TWiVo in a museum, and we were walking around with dinosaurs everywhere. Yeah. And I said, Nels, there should be a museum with big viruses everywhere, big models. Wouldn't that be cool? You kids would love it. I would love it.
Well, the American Museum of Natural History has a hand-blown glass section of protozoans. They're quite
That's nice.
Elegant.
So I figure a room with variety of different. So if he's willing to work with us, I'll tell Nels, and that would be fun. Then we could go and do a TWiV right there.
You bet.
Dickson, you're next.
Brian writes, TWiVome. Greetings from Allentown, PA, with a wintry mix and 1 single degree Celsius. Firstly, I would love to receive the Emerging Infections book because I hear they're really going to catch on. Secondly, I think I may be eligible for a TWiV coffee mug, having submitted 2 haiku poems to the contest. I believe there is no upper limit to the number of Twixt-related mugs that a person should own. Finally, I would like to express my sympathy to Vincent and pass some advice on to the audience, which may help to limit his distress at having to keep the book contests running for such extended lengths. Jesse Thorne of NPR's Bullseye and the Judge John Hodgman podcast had some helpful tips for anyone who is working their way through back episodes of their favorite podcast. To wit, he recommends listening to the podcast in order, but interrupting that flow to listen to the latest episode when it comes out. This allows a fan to keep current regarding upcoming live shows, promotions, or contests, while also getting to appreciate the full catalog of content. I strongly suspect that the lag in submissions is connected to our desire to savor the collective wisdom of the panel and your guests. If my DNA extraction load had been less heavy recently, I would likely not have spent enough time at the bench to become current once again myself. Please keep up the excellent work. Best, Brian.
That's exactly the approach that I used when I started listening to TWiV and the back catalog. And it worked okay, but then when some topic would come up, I couldn't remember whether it was really recent or whether it was 2 years old. Right. Not that it really mattered much, but I wanted to know whether I was current or stale. So.
Oh, sorry about the mug, Brian. You have to send me your address. Just email it.
Yep.
So that because I might not remember to ask you. And I will send you a mug for sure to those Limerick contest entrants. All right, Kathy.
Stig writes, hello all. The weather here in Denmark is cold, not using a weather app. All I can say is that it's very dark and cold when I bike my son to the nursery on my way to the local hospital. I just started listening to TWiV episode 428, and I heard that you had not yet received the 27th email for the book. So I would like to enter the contest for a chance to win again. This is my second entry for a chance to win the book Infections of Leisure. I felt I needed to clarify that. I would like to submit what I think is sadly a very necessary pick, although not related to science. It's John Oliver's Last Week Tonight called Trump versus the Truth. Season 4, Episode 1. Sincerely, Stig, keep up the interesting conversations. So, and he has a YouTube link to John Oliver's.
It's pathological.
Yeah.
By the way, I wanted to mention that this is episode 434.
It is.
And there is a Lambda phage called Lambda 434. And it's just been, ever since I started the show notes, it's been in my mind, this Lambda phage.
Houston, the Lambda has landed.
Lambda 434. All right, let's do some picks of the week. Hey, Dickson, what do you have for us today?
I've got a pick. Let me scroll down here just to make sure I remember what I picked, but I think I know what I picked.
You said, wow.
I picked Vicious Animals. And these pictures are astounding. And, you accuse me of just paying attention to Yahoo News. I use that as a foil to get my laughs every day because a lot of it is fake news. But sometimes you come across these photographic essays, so to speak. And this one, it claims fierce animal portraits, but there are some non-fierce animals in there too, like the gorilla pictures don't look too fierce, and some of the chimpanzee pictures don't, but they capture they're almost anthropomorphic the way you look at these animals. They express human emotions through their faces, and I think that's photography working there, by the way. But I really got a really interesting perspective from these close-ups of these animals. I really loved them.
They are really nice
They have
pictures.
Stephen Hawking every now and then.
I don't know. But you can't get rid of the ads that come into.
But there are animals, all animals, but then they have him now and then.
Yeah, and then have a candy bar and something else. But that's Yahoo News for you. Sorry about that.
Flamingos are cool. The bat is really neat.
Yep.
That chimpanzee's giving us the finger.
Yes.
Is he?
That's exactly correct. Flying the bird, as it were.
Yes.
Oh, yeah.
Giving the camera a hand gesture is the caption.
Correct.
Dickson, you didn't get this at Yahoo? Is that what you're saying?
I did get it at Yahoo. I did. It's useful.
All right. Thank you, Dickson.
You're welcome.
Kathy, what do you have?
Well, I picked a story about the Bodie McBoatface submarine. And so you may recall that there was a story about a research ship that the British government asked people to suggest names for. And when you do something like that on the internet, you get what you ask for. And Bodie McBoatface was the winner. And the second place one went to something called RRS Henry Worsley. But in the end, the government decided not to name the ship that, but rather the RRS Sir David Attenborough. And the radio guy, the BBC radio guy who started the Bodie McBoatface meme, apologized and said that he voted for the Sir David Attenborough name. Anyway, they decided to go ahead and name a small submarine, or maybe 3 small submarines, Bodie McBoatface, after all. And they are launching now for one of them. The first one is launching for an Antarctic expedition. And so this is the link to that. And it looks like it's about 15 feet long.
It's
maybe.
a yellow submarine, basically.
It's a yellow submarine.
and since we all live in a yellow submarine.
They describe what it's going to do. So I thought it was kind of fun follow-up story. Kind of did the meme of the science, exploration, and the Antarctic, which we've covered before. And then I also wondered about the Bodie McBoatface meme, or the phrase or whatever. So I looked up a little about that. And it has another possible name, or they say it fits into a category of something called a snow. Oh, shoot. I emailed it to myself. Now I can't find it. So the phrase "orange is the new black" is another one of these. Though just various kinds of phrases. So it fits in the looking up words and meanings and phrases and things. So the Bodie McBoatface or X McX phrase is this theme. So
nice. Alan, what do you have?
I have a pic from, I think I picked the Bug Chicks a while ago, but this is a video that they did. They actually did this video about 5 years ago, but I only recently discovered it. This is a parody video from a movie scene from the Star Trek The Wrath of Khan movie. And they are correcting a very common myth about an insect, the earwig. So earwigs, people think, oh, it's called that because it crawls into your ear at night. Ew. So they spoofed that, and if you're familiar with The Wrath of Khan, then you'll recognize the scene, and you can probably predict how this goes. But a very effective science communication and very cute. So
there are bug chicks because they're entomologists. Is that correct?
Yes. Yeah, they are. They talk about entomology. That's their shtick.
So this is a bug podcast. No, it's not a podcast. It's a video, right?
It's a video. This is a video from them.
I got it.
They do a bunch of videos. They've done some books. They've done a bunch of other things. They're very cool.
It's pretty funny.
The ladybird beetles.
They focus mostly on like K through 12 education level, but a lot of fun.
Yeah, it's funny. Dickson, you know what an earwig is?
You betcha I do.
Do you think they have viruses?
Oh, everything has a virus.
Of course.
Even viruses have viruses. You taught us that.
That's right.
The Mimi virus has a virus.
Well, it's a virus of a virus-infected cell.
It's a virus of a virus-infected cell.
All right. Now,
I looked up my phrase, snow clone. It's a cliche and phrasal template originally defined as a multi-use, customizable, instantly recognizable, time-worn, quoted, or misquoted phrase or sentence that can be used in an entirely open array of different variants.
Okay. Give us another example.
Well, so I said, orange is the new black.
Oh, I see.
X McX, the mother of all wars, the mother of all this, that kind of thing. So those are all snow clones.
Snow clones. Right.
Cool. All right, my pick is a little unusual because I'm not a big TV fan, as you know.
No, you're not.
But my pick is a TV show.
Get out.
Called Westworld.
I know that show.
Now, the reason is last week was spring break. All my kids were home, and my older son, Aiden, comes home, and he says, Dad, you have to watch Westworld. So we watched like 2 episodes a night. There are only 10 first season. And I found it very engaging. Now, it is violent, and
yes, it is.
I found that a little upsetting. I just don't like violence, but because it upsets me. But the premise is fascinating. Has anyone seen it?
Did you see the movie?
No,
I saw the original. I saw the original movie. The
I did
73 movie.
That might be interesting to look at. So this is a remake, basically, but in a TV series.
It carries
format.
it forward, right?
And very interesting. You know, the premise, it's a Western theme park, essentially, where the hosts are all androids, basically, that look very human, but they're mechanized inside, and they're controlled by the owners. So they go through things every day, you know, so they get up, and they can be shot, they can be assaulted, they can be done anything with the guests who come in and pay $40,000 a day to be in this park. And, you know, the violence, I think, is not even needed, but the intricacies of what's going on are just amazing. And things happen that you have no clue about. Do you want me to tell you all the spoilers?
Go for it.
No, I would never
We're
do
not going
that.
to watch.
I would never do that. But it's quite interesting, the whole idea of, you know, controlling an android from a remote computer, which they do all the time. You know, so they interview their androids they're called hosts. They interview the hosts, the people who are running the thing.
Right.
And they'll ask them questions, and if they go through their usual emotional routine, they'll go, cut out the routine, straight answers, please. You know, and then they'll just give them the facts. It's very interesting. So I thought it was cool, and I watched all 10, and the last one was very interesting.
You binged.
And, 2 a night, and I think it's really cool, and it's science based because it's all about making artificial people and controlling them. But in the end, it's really about how crappy humanity is.
You think?
Yeah, because people go to this park just so they can shoot people and have sex with them and have no repercussions whatsoever, and they do it. And even people who go in and say, "No, I don't want to take a gun," they have the hosts have all these little routines that they're programmed to do. They're storylines developed by the programmers. They change from time to time, and the people they come in and they see something happening and they jump in and participate. And there's a moral story to this in the end, and that people are pretty nasty, and that's kind of sad. Did I need a TV show to tell me that? No, I didn't. But I'm sure a lot of people have watched it, and it's quite interesting. I would check it out. But what they do with modern TV is not like the TV I used to watch when I was a kid. Boy, it's so different.
Oh, yeah.
An episode is almost like a movie.
That's right.
By the way, there are 10 episodes of this series. It cost $100 million. It was made by HBO. And what I could do with $100 million for microbe TV. Wow. Of course, nobody would watch it, I guess.
That's not true.
No. Yeah, I bet if we produced a $100 million show, it would be one that people would watch.
Oh, they'd watch.
Well, we could do $1 million shows. How about that?
That would be nice.
Those would be pretty amazing, too, I bet.
We could do it even for less. We'd only need, $10,000 per show just to do some graphics. We wouldn't, videos.
At $1 million a show, we wouldn't have to pick papers. We could fund them.
Yeah,
exactly.
Right.
We could write them. Are you kidding?
That's right. We have another listener pick. So we had that last one, who I don't remember, but Hannah in Boston. My listener pick is the mobile game Plague Incorporated, Plague Inc. It's a fun, if scary, strategy simulation game where you control a plague in an attempt to create a global pandemic. You can play as a bacteria, virus, fungus, parasite, prion, or even a fictional pathogen that turns people into zombies. My one nitpick is that the prion plague type includes DNA. Is it so much to ask for a little realism in a zombie apocalypse game?
Right.
I think it's too much. But I agree that they shouldn't have DNA in the bloody prion. That sounds very cool. Plague Inc. Dickson, you remember when we did an episode with Aiden about viruses in video games?
Way back when.
Like number 7 or something like that.
Back in the day.
That was when we never knew if it would ever finish.
No, we did not.
All right. Thank you for the listener picks. You can find TWiV at iTunes, microbe.tv/TWiV, parasiteswithoutborders.com. I'm going to see if you're listening.
I'm listening.
Where else can you find it? How do you get it on your smartphone?
I don't have a smartphone.
How do you get it on your tablet, which you have in front
You
of
type out microbe.tv/ and then you put the right...
You could do that. Or you could get an app that plays podcasts.
Or you could get an app that plays, right? Sure.
Just subscribe in those. And on your...
And it's all free. It's all free.
There's one called Podcast.
Really?
Subscribe. Folks, just subscribe, please. We'd like to have billions and billions of subscribers.
We would.
All right.
And you can all write letters for books.
And if you'd like to help us out, and I do appreciate, 1 or 2 persons a week, help us out. Go to microbe.tv/contribute, and you can try our Patreon account. You can you can buy things at Cafe Press or Amazon, or give us cold hard cash, and it will help us to do some of these cool things that we're talking about. Right? Dickson Despommier can be found at parasiteswithoutborders.com, where you can get a free copy of Parasitic Diseases, 6th edition.
It's true.
Right? And
new second printing.
New second printing.
It's true. Just put it up last week.
Less than I put it up, dude.
I
put it up.
I mean, you're part of the team.
I put it on the website. That's what I mean by I put it up. You guys wrote it. I didn't have anything to do with the writing.
No,
but it's a royal we. Yeah. That's fine. For which I got a penny. And Parasites Without Borders. But at thelivingriver.org, there's nothing free there. Just some pictures and writing, right?
That's true.
Oh, that's a lot. That's a lot.
But you can get something for 99 cents, for gosh sakes. I mean, come on. You can download the Duets book for $2.99.
Oh, that's right. You do have some books there that you can buy very cheaply.
Waist deep in water.
Has anyone bought any? Have you checked?
Not too many.
I'll buy 100. Okay.
Oh, thank you.
Make you feel good. Thank you, Dickson.
Great. You're welcome.
Kathy Spindler's at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thank you. This is a lot of fun.
Alan Dove is at alandove.com. He's also on Twitter as Alan Dove, no underscore, because he's the original Alan Dove. Thank you,
That's
Alan.
Right. Thank you. It's always a pleasure.
Just love underscore Dickson. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this episode, Blue Apron. The music you hear in the beginning of TWiV is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. I don't know if Alan said he might be late.
Yeah, there might have been a few little flaws in this paper, but I thought that the paper was interesting from some perspectives. So secondary sexual characteristics, you know what those are, right?
Yeah, but it has nothing to do with insects.
Why not?
Well, and I think it means something completely different. Or I think secondary is just a gratuitous word that they threw in.
Yeah, I think the writing is really...
Well, if they don't mean secondary sexual characteristics, then they are wrong.
Yeah, I don't think they do. Now I can't. Where did I...
Because if you can see the difference between a male and a female wasp, and you should be able to see the difference, then those are the secondary sexual characteristics.
Right. But what they're saying has nothing to do with that.
I agree. I think we could be critical because there are a lot of issues I had also. First, I didn't understand half of the words in the frickin' paper.
Well, let's go through that, shall we?
Oh, yeah.
I might be able to help you out here.
Yeah.
I don't know what...
yeah?
Hold on.
Yeah, I know the word you're looking at. I know where you're going.
I do too. I had to look one.
It's got something to do with insect development.
Yeah, probably.
Yeah.
What is a supernumerary egg?
No, that I don't know.
Supernumerary is like
More than normal.
more than expected. Yeah.
More than normal, that's all.
They give it an adjective like that?
Well.
Yeah. Supernumerary. There's like supernumerary stars or something.
Okay. What about...
Too many stars in Hollywood.
Hey, Dicasimimorpha longoicada rhabdovite. What the hell? I can't even pronounce it.
Got some cool names.
Supernumerary.
What's a cruciferous vegetable, Dickson?
Broccoli.
Cauliflower.
What's a synovigenic parasitoid?
One that keeps producing eggs through the adult stage.
Look at that.
That's the one I had to look up.
Look at that.
Okay.
Yeah. But the vitellogenesis I didn't have to look up. That's the formation of yolk.
Correct.
I don't know anything. Unambiguous.
No, you just don't know any. Yeah.
Unambiguous viruses. That's the title of our show.
Unambiguous virus.
They have, that's their conclusion. We have unambiguously identified.
Supernumerary teeth. Presence in excess of the normal or requisite number of teeth. There are supernumerary
fingers.
What's the other? Yeah.
They have 6 instead of 5.
But it has one more syllable than I thought. Supernumerary digit, supernumerary nipple.
Yep.
Yeah. So more than expected.
Can you have supernumerary intelligence?
No.
No, I think it's a counting thing, not a.
It's numbers.
Yeah, that makes sense.
Thing.
Numerary, super, more than. Yeah, got it.
Right.
You can learn a lot. On TWiV, you know.
The language of entomology is strange to begin with. It's got lots of anachronistic expressions and.
Dickson, you are familiar with it because I
listen to mosquito biologists a lot.
Deal with people that study these because your parasites are often transmitted by them, right?
That's exactly right.
I appreciate that. Now, do you think if I worked on an arbovirus that I would also... But Kathy worked on an arbovirus, or maybe that's why she knows these words.
Yeah, but maybe, but if you didn't work on the arbo part of it, no.
But, Kathy, when you worked on VSV, did you think about the vectors at all?
No, I don't think I even really appreciated that it was an arbovirus.
Yeah. All right.
Yeah. So the secondary sex ratio alteration, the underlying mechanism of secondary sex ratio alteration remains unclear. So they're not talking about secondary sexual characteristics.
No. No, they're not.
So they're talking about something else, and I think it's just a gratuitous word or something that didn't translate right.
Who handled this paper?
Well, it started out to be Jens Kuhn, and then they enlisted him to be an author.
Francois Balou. Oh, where did you read that?
On page 2 of 27, the left-hand column.
Right.
So you can be an author just because you can translate Chinese into English?
Well, no. He was initially assigned as editor, and then after revised decision, they contacted him to collaborate on the revision due to his expertise.
I see.
Yeah, the editor was Francois Balou. What does he do? Francois Balou. Let's look him up.
You know, maybe there'll be some insect. Maybe
Alan knows a lot about them.
He knows about guinea worms.
No, he knows a lot about insects because his grandfather was an entomologist.
Francois Balou is a computational biologist.
That doesn't
help. No wonder
doesn't help.
They used gratuitous
He's a supernumerary biologist.
Oh, my gosh. My primary research is to use genomic data to answer fundamental questions in biology and medicine.
Oh, that sounds pretty generic.
I railed against computational biology.
Railed, that's right.
Do you know what that word means?
Yeah, I got pushed up against the rail. I came in third in a 2-horse race. That's difficult, but nonetheless.
AD may be a few minutes late. All right, we're more than a few minutes late now, right?
We are.
6 minutes.
Yeah.
My clock is ticking here.
We can start. Maybe he'll get here by the time we're done with the weather.
Sometimes people say just a few minutes to mean like an hour.
I know.
And in Italy, they do that.
And in Egypt, they mean like, I will see you tomorrow.
And one thing, when I was a kid, we used to go to Italy. And my parents always complain, you ask somebody directions. They say right up the street, and it's 30 miles away.
That's right. Well, it was right up the street. What do you want from me?
Oh, here he is.
Uh-huh.
Okay. Our supernumerary podcast.
That's right.
Podcast.
Are you supernumerary, Alan?
Well, I hope not.
That's right.
And what would you interpret if I said that?
That I had too many of something.
Exactly.
And if you had a podcast called the Supernumerary, which nobody could pronounce, podcast, what would that mean? One too many?
One too many podcasts.
There you go.
We figure you're going to explain, you and Kathy are going to explain all the words in this paper.
Well, the main one, Alan, the main term that I just think is wrong is the secondary. Where is it again? The secondary sex ratio alteration.
I don't understand either.
Oh, come on.
Okay. I think it's a gratuitous throwing in of the word secondary or something that didn't translate well in Google Translate.
Yeah. Yeah, because I mean, I know what secondary sex traits are.
Yeah, that's what I thought too. But,
but doesn't seem right here.
All right, good. We can
maybe ask the authors to clarify.
Perhaps we can come up with a good ambiguous title for this.
Well, good thing I only picked one paper, right?
Yes.